Okay, thanks. So, yeah, thanks first of all for having me here. It's for me also a very special meeting, I have to say. I will be talking about a little bit about things that go a bit beyond just cell-cell interaction, but that's also stuff that we do. Just maybe to start introducing myself a little bit. So the title of the talk is Microbial Nitrogen Cycling Networks in the Changing Ocean. Something that I'm lately very interested in. When I say I'm part of a bigger department. I don't do much work myself anymore, but in that department we work on mainly nitrogen cycling, but also on trace greenhouse gas cycling. And what we're interested in is anything from at a single cell level, looking at microorganisms, mainly working on oceanic environments. And what you see here is a drop of water that you can filter on a drop of seawater that you can filter on a filter, and that's what you get. That you get a whole bunch of bacteria that all look the same. They look actually pretty boring, to be honest. Then we look at what they're doing, how they interact with each other, and here's an example given from a haptified algae that lives in symbiosis with a special cyanobacterium that is not capable of doing photosynthesis, but provides its host with fixed nitrogen because it can do nitrogen fixation. And we look at how these organisms actually, what their role is in ecosystem function, and I will come to that quite a bit in my talk, but also on the cycling of carbon and nitrogen in particular. And you can imagine how with a picture like this, where you can see a bloom of N₂-fixing cyanobacteria, you can easily imagine how this impacts the biogeochemistry or the biogeochemical cycles of carbon and nitrogen in this particular case. In the end, what we want to get to, however, is to see how these processes affect the chemistry of our atmosphere and ocean, and why the ocean, I think, just looking at this Galileo spacecraft going away from Earth, making a picture of our planet. If you look at our planet, it's actually an ocean planet with a little bit of land on it. I mean, it makes a picture just by chance, and what you see is water. So oceans are important, but why nitrogen? That's probably a question that is not. Oh yeah, and before I start with that, I should point out there's quite a bit of unpublished data in there, and this is from the people that have been producing that data. Let's see what goes best. So why nitrogen? I said, so well, nitrogen availability controls growth in the ocean, and actually it's estimated that more than 70% of the ocean is nitrogen limited. And what I'm talking about is the ocean's surface waters. If you would add nitrate or ammonium or nitrite, so bioavailable nitrogen, to these environments, you would stimulate algal growth. And you can actually already see that from the data that you have here, where you have in the top chlorophyll concentrations that are measured from satellite. This is, I think, an annual mean, if I remember right, where you can see the chlorophyll distributions, high concentrations up here in the north. You have this in these equatorial upwelling regions, and also here in these eastern boundary current upwelling regions, you see high chlorophyll concentration. I'm talking about upwelling. That's where water is coming up, where it's being brought up, and what it brings up is nutrients. And nitrate is the most common form of fixed and bioavailable nitrogen in the ocean. And you can see that particularly here in the Northern Ocean, you have a very good correlation between nitrate availability and chlorophyll concentrations. The same here in the equatorial upwelling. So nitrate is important. It controls the growth of algae, and thereby it also controls the uptake of CO2 by our ocean, because algal growth photosynthesis is that what drags down CO2 and drives the carbon pump, something that's very relevant for all of us. And so if you want to understand, well, the carbon drawdown is maybe for us from a human-centric view, the most important thing. If you really want to understand that, you have to understand what fuels that system, and the fuel that drives this system is nitrogen, bioavailable nitrogen. And we try to study basically all processes that modulate the availability of bioavailable nitrogen, so nitrate, nitrite, and ammonia in particular. And I just give you an overview of the various processes and with the fluxes associated with it. And actually the only home-taking message from this, or only message that you have to take back home from this, is actually something that the nitrogen cycle doesn't exist. there's separate cycles because the magnitudes of cycling are very different between the different processes. Like you have processes like, for instance, the nitrification that was mentioned before, where ammonium is being oxidized to NO and then via nitrite goes to nitrate, which is a very big process with 2,000 teragrams per N per year being turned over, while another process, N₂-fixation, is only turning over 180. Already from that, you can see it cannot be a balanced cycle. It's several cycles that are working in different modes and different time scales. However, processes like this, nitrogen fixation, are vital, even though it's turning over not that much nitrogen, because actually until human activity started changing and perturbing our nitrogen cycle, the only way to get new nitrogen into the ocean was actually by fixing nitrogen. And it's still the main source of new nitrogen, fixed nitrogen, bioavailable nitrogen to the ocean. So there's a few organisms, bacteria, archaea, that can convert atmospheric nitrogen, the biggest pool of accessible nitrogen, into something that is bioavailable for all others that cannot convert dinitrogen gas. So I will be focusing mainly during my talk on this. As I said, we work on all these aspects, but that will probably take too long. So we work on nitrogen fixation, and in particular what we're now interested in is also what is happening to the nitrogen fixation and in general to the nitrogen cycling due to global change issues. I mean, I already said until not that long ago, until the industrialization, or actually the invention of the Haber-Bosch process that converts atmospheric nitrogen with high temperatures and high pressure into ammonium, the only way to get nitrogen actually in the ocean, or the main way to get nitrogen in the ocean, was nitrogen fixation by organisms. This added nitrogen has had horrific effects on our environment. Adding all this fertilizer nitrogen has done quite a bit, and particularly to the coastal regions. And one of these ecosystems that is very strongly impacted by that, and also by other factors, by the way, is the seagrass meadows that you can find in many areas around the world, from the moderate climate regions where we are now to the tropics. They're everywhere, basically. They can make this very dense communities of plants. We like here, this is Posidonia oceanica, and I will be talking more about it. That is a seagrass that you can find in the Mediterranean Sea. And that plays a very important role as a nursery and food place for many animals. And that's for us relevant because actually quite a few, I think out of the top 50, quite of the most important catch fish in the ocean, many of them have hatcheries actually in these seagrass beds. If they disappear, these fishes will disappear. So ecosystem loss is very important to us because it has also economical consequences. Food is disappearing. But beyond that, and that's what we are interested in, it also has impacts on the global cycles of carbon and nitrogen, or the global cycling of carbon and nitrogen, I have to say. So, and that's what we're interested in. But before we start that, we first have to understand the system to a certain extent. And that's what we're doing. We're using all kinds of experimental methods to look at, in this case, nitrogen fixation as a source for seagrass growth. So it has been hypothesized already for decades that many of these seagrass meadows are fed by N₂-fixers. And why is that? Because if you look at a system like that, then you see such a nice high biomass system sitting next to something like this white sands in pristine blue waters. And blue waters means there's very little nutrients in the water. It's not only there's no DOM, there's no nitrate available, because if it would be there, then there would be algae and they would take over. It's not the case. So actually there are some algae growing here on the sands. And if you look at their productivity, and you can use a method that is called eddy correlation flux measurements, where you can then determine the oxygen flux, and that you can directly convert to CO2, because photosynthesis in that respect is relatively simple. You can look what is happening in these sands. And if you look in these sands, then you see actually they're not really fixing CO2, they're not releasing oxygen, they're actually releasing CO2. So these diatoms are not doing very well over there. And this is in sharp contrast to the seagrass, because this seagrass bed, which we see here, has massive oxygen fluxes and CO2 fluxes associated with it. And how big is this? Well, the fluxes that you have here per square meter is equal to that what you get in a square meter of pristine rainforest. So it's massive. Well, how is that possible if there's no nutrients in the water? And that's why people said probably N₂-fixes. What was said is like N₂-fixes might be growing in the sediments, around the roots. They might be growing, and that is a hypothesis that came up some years ago in association with mussels that have them living in the gills, and these organisms die off, and the nitrogen is released and feeds the seagrasses. We, however, were thinking maybe it's a bit more direct, because actually the seagrasses are land plants. They moved from land to the sea 100 million years ago, roughly at the time that the angiosperms, the ancestors of these plants, evolved. So they moved back, and what we see on land is that many angiosperms actually that live in a nutrient-poor environment have symbionts associated with them. The most famous one is the legumes that have rhizobia in their root nodules. So we were wondering if something like that might be going on here as well. So what we did is we took these seagrass plants, put them in a pot, put some a rubber membrane over it, locked it off, and then added a tracer to the seawater. And what kind of tracer? We used a rare isotope 15N from 15N nitrogen. Most nitrogen is in the 14N form there. So if you add 15N2, you can start following it with mass spectrometry. And then we had in the upper part, we have you can see the plant, the leaves, and they were in an aquarium just growing without tracer. Then we looked and incubated many plants, and you can see the plants over here, or doing seasonal studies where we looked what is happening. And we could actually, in April and May, we didn't see any N₂-fixation happening in these roots. Nothing is happening, which fits because actually in those seasons you have small amounts of ammonium and nitrate in the waters there. However, then in summertime, in June and August, we can see high rates, and these are really among some of the highest rates that we have ever measured for nitrogen fixation happening in the roots of these seagrasses. So I should tell, this is all sediment free. This is water, this is just the roots and the rhizome of the seagrass in the pot. But we do not only see that there's nitrogen fixation happening in the roots, we can also see that this nitrogen is actually being transferred to the leaves, because there's no tracer up here, but still, if we combust the leaves and measure the isotopic composition, we see that nitrogen has been transferred. And how much? Actually, it's more than enough that what is being fixed here and what's just being transferred to sustain the seagrass growth during the summer months. And that's very important because actually the summer months is the main growing season of seagrass. So basically the seagrass meadows, to a large extent, are supported by nitrogen fixation, by these organisms, wherever they might be in the roots. So then we wanted to know, who are they? And for that, we used the classical amplicon sequencing approach to first try to identify the organisms. So what you see here in the bottom is the amplicon sequencing of seagrass root pieces, and in the upper part you can see the associated N₂-fixation rates with it. So here, when you have little to no nitrogen fixation, you can see already something coming up that I will be talking about quite a bit now, is Salarina tantimonus. It's a bacterium that is there always basically in all roots that we looked at, but at very low abundances. However, that condition completely changes in summer when we have high N₂-fixation rates. And then we see this one bacterium, it's a single species that starts growing up in summer and makes up, just based on the amplicons, up to 60, 70, 80%. In some of these roots, and particularly in the roots with high N₂-fixation rates. So we have our candidate, can we then now link it? So and where are they? That's the next question, because what we were thinking, maybe they're living on the outside of the roots, they provide nitrogen to the organisms. Then we looked and we designed a fluorescence in situ hybridization probe, and you can see the results of that here. So the blue, purple, and orange colors is the organisms that we had targeted with our multi-fish probe approach, where we use several fish probes to visualize the bacteria. The green stuff that you see is actually the plant material. And for those of you who are familiar with plants pieces, you can already see that we're not sitting on the outside of the root, we're sitting actually on the inside of the root. And then we can do something nifty, and that is a method that we use quite extensively in our institute. We have a system that allows us to analyze the isotopic composition of a single cell. And that's called NanoSIMS, nanoscale secondary ion mass spectrometry. What you do is you bombard your sample with primary ions. These ions disrupt the surface of a sample. As a result, atom clusters and ions are being thrown out, ejected, and some of them are ionized, and they are the so-called secondary ions. And they give you information over the isotopic and elemental composition of the root material. If you do that, then you can make an image like this. You can then map the 15N distribution in these in the cells as well as in the root pieces, and then you see that the highest abundance of the 15N label coming from 15N2, because these are from the same incubation spots that I showed you before, is ending up in the bacteria. But also the roots are enriched, because black actually in this case is no enrichment whatsoever. So there's a transfer of nitrogen from Cellerina tantymonas, because it's a specific growth, to the roots. And we can directly show that. Then what makes it very interesting to us is the question that we were, so because it says like terrestrial type N₂-fixing symbiosis between a marine bacterium and seagrass. Well, if you think about it, it's not directly logical. You have a plant that comes from land, that has evolved on land, moves back into the sea. In principle, it could bring its own symbionts from land. It could bring a rhizobia down, but it doesn't. It's actually a gamma proteobacterium that has nothing to do with the rhizobia. It's actually closest related to seaweeds, bacteria that grow on seaweeds. So, however, the system that it uses is very reminiscent of that what you find in rhizobia. So what it does, and it's a simplified sketch based on molecular data and also experiments that we did, we could show that the Cellerina tantymonas fixes nitrogen, provides ammonium, but also amino acids, just like rhizobia, to its host, and in return gets, in this case, sugars. That's a bit different from what you see on land, because on land often it's acids that are being transferred. Then the plant, of course, gives the sugars via photosynthesis. So said marine bacterium that behaves like a terrestrial symbiont. And actually, what was very interesting is that the adaptations that this marine organism needed to make to live together with this plant is probably not so extensive. If we look at the organisms that we can find on these macroalgae, these agarivorans, it has quite a few things in common. Actually, basically, the genomic differences were relatively minor. Or both of them are basically able to do nitrogen fixation. We believe that because both are probably growing on sugars, also macroalgae are sugar-rich environments. And the main step actually to get into the seagrasses, so that came later, 100 million years ago, while probably these macroalgae were already around for a long time before that. What they needed to do is get into these plant cells. And for that, they got something special. They have a pectinase that is not present in any of the other relatives of them. So they have picked up something that helps them to degrade a cell wall of the plants and of the land plants, terrestrial plants that have pectin in the cell walls and could by that move in. So now what we're moving on to and looking into is, as you see, for those of you who have been carefully looking, there's many other relatives that you can find of Cellerina tantymonas neptuna, as we called it, in other plants, in other seagrasses like Thalassia imbrici, but also in cordgrasses that you can find in salt marshes. And we're looking if they play a similar role there. I move to another topic and another project, which at first might not have much to do with nitrogen, but I will show you it has. And that is actually the substantial methane emissions from seagrass meadows as well. That's also well known. That's already known for decades. How it works is not completely clear, because although it might make sense at first sight, it doesn't. You have these seagrass meadows that have peat deposits under them. I will show you some pictures of that later on. And these peat deposits are anoxic. You can measure oxygen concentrations as we did it here, and you can see that like after a few centimeters, these sediments are anoxic. Good environment for methanogenic organisms that produce methane. So, and indeed, methane is there at substantial concentrations, considering that it is an open porous sediment where there's continuously flushing out of material as well, at a low space, but still. The problem that methanogens run into, which is different from a peat on land, is that here you have tons of sulfate being present. And sulfate is a bitch for methanogens, because sulfate reducers outcompete methanogens for their favorite substrate, acetate and hydrogen in general. So, and we can see that sulfite is being formed, so sulfate reduction is happening. So how can we then have methane build up, and we can measure the metal production in this system when there is sulfate around? Well, these competitive substrates in acetate and hydrogen, we tried indeed, we do not see any methanogenesis happening with them. But if we give them non-competitive substrate, and that's methylated compounds like methylamine and some other compounds, then you all of a sudden see high methane emissions. And why are these compounds irrelevant, and why is there a link to the nitrogen cycle? Well, actually, these compounds that I'm talking about, the nitrogen compounds, these methylated compounds, most of them are derived from betaines. So the breakdowns from betaines, and betaine is a osmolyte that the plants needed to produce in huge amounts to cope actually with the fact that they're living in a saline environment. So this feeds the methanogens. As you see, it's nitrogen containing, and the nitrogen, as I said, in this system is mainly coming from nitrogen fixation. So there's a direct link between the nitrogen fixes providing nitrogen to make these betaines and methanogenesis happening. There's a pretty detrimental part to this story as well. We studied the methane emissions from the living seagrass, and we also looked at dead seagrasses. There's a dead seabed, dead seagrass bed close by the patch we were looking at. It's already gone for 20 years, 20, 30 years. And we measured the methane emissions from them. They're identical. There's no difference. And the reason for that is very simple. The rhizomes and roots that are there, that are left behind, contain still huge amounts of betaine and breakdown products of betaine. And they keep on continuously releasing methane. So that brings me then to the end of this part. And it is that we try to actually see what the impacts are of the disappearing seagrass, because that, I said already, seagrasses are disappearing because of nutrient loads, but not only that, we're doing dredging for sand, there's anchoring. If whoever has ever gone to the Mediterranean, for instance, Mallorca, I was recently, you see always these nice round patches or nice round holes in the middle of seagrass patches. That's because people throw out their anchors, pull them up and pull the seagrasses out. They're on the decline, they go very fast, extinct as it looks like now. There's predictions that by 2050 there will be no seagrasses in the Mediterranean anymore. And that will have impacts. So it's believed that they play an important role in blue carbon sequestration, so they play a similar role as burying peats on land. And you can see why if you look at this picture. This is of Corsica, the coast of Corsica, where you have this thick peat deposits formed by Posidonia oceanica. At the same time, these peat deposits are producing methane continuously. So this particular one is died off. It's not really doing any blue carbon sequestration anymore, but methane is being released continuously from it. So we think this has an important impact on the carbon cycling of the ecosystem. Globally, honest to God, I'm not sure. What might be more important actually for the whole ecosystem is actually the N₂-fixation. The whole Mediterranean is now nutrient poor. So the seagrasses have very high N₂-fixation rates, and we're convinced, or we're sure, that the N₂-fixation happening in these roots is driving the ecosystem as such. So the coastal ecosystem, but maybe also the open ocean system. And that's something that we're currently looking into, not only in the Mediterranean, but in general in oligotrophic environments. Who is responsible for the nitrogen fixation? Is it this kind of coastal environments that leak out nitrogen to the open ocean, or is there also nitrogen fixation happening in the open ocean? Not that this is not known that there's N₂-fixation happening, but there's some open questions there that I at least find difficult to answer for me. And this is, if you start looking, one of the projects that we're working on is the nitrogen fixation in the ocean deserts. Already Jeff has been talking about these oligotrophic systems where you can find Prochlorococcus, and they're generally considered ocean deserts. And why? Well, not because there's no water, there's more than enough water, but vegetation, if you want to put it that way, is low. There's low productivity. And you can see that again from the chlorophyll map. You see these black holes in here, and that's actually where the satellites cannot detect chlorophyll. Chlorophyll concentrations are low, and the general idea is, yeah, there's no nutrients, bioavailable nutrients around there, but this is also the ideal environment for nitrogen-fixing organisms. The question that I always ask is then, but why don't the N₂-fixing cyanobacteria that can do photosynthesis and can take nitrogen from the atmosphere, why do they not fill this hole with chlorophyll? And the answer that you generally get is because they're limited by something else. And something else, what people talk about, is actually iron. And so what we started looking into is the actual impact of iron on N₂-fixation. And where we went to is the tropical North Atlantic, because that's actually the environment where there's a lot of dust input coming from the Sahara. I don't know if you can see it. There's a lot of clouds, but you can see maybe here this brownish gray belt going all the way from the Sahara to Mexico, basically, and depositing dust, which contains iron. And this is an environment where you indeed, if you start looking at it, find some of the highest N₂-fixation rates that you can have. And what we try to do in an experiment that many people have been doing before us is adding iron to the seawaters, adding 15N2 and looking at the N₂-fixation rate, so the uptake. So you look at a control and a plus iron and then compare them. And if you do that here at station 4, it doesn't matter where it is, they're all close together, you see there's actually no significant effect. Nothing is really happening. But at other places, and that's what got us excited, oh, look at this, here we add iron and N₂-fixation rates go up. It fits to the model that we have. We should have stuck to looking at bulk samples, but then we started looking at the single cells, and then everything starts to fall apart. And why? Because it's not one organism that is found in these waters. You basically find all N₂-fixing cyanobacteria that you can imagine are growing there in parallel. And they respond differently, and that's, I'm giving away a little bit of what I'm going to talk about next. But if I talk about all, I just give you an example. This is a picture, I think, from the hot station, from Rachel Foster, who was a postdoc in my lab in the past, where she found different types of Trichodesmium. That's a filamentous N₂-fixing cyanobacteria. It's not so important who it is. Then there's these diatoms that have symbionts in them or on them, like this diatom hemiaulus that has Richelia as a symbiont. Then you have Rhizoselenia, another diatom that has also Richelia, but another Richelia strain as a symbiont. Then there's this single-celled Crocosphera. They're all there. And our question was, actually, and naively, we started out who responds when we add iron? Who is responding in station 7? Station 4 we just took as a control station. But actually, I would like you to focus on station 4 because that's actually the most interesting result. I will come to that in a moment. So first, we again use NanoSIMS. We reveal that which reveals differences already in background activity between N₂-fixes. So you have this Trichodesmiums, you have Gunadia that keep on renaming the diatoms associated with the cyanobacteria, but it's one of these diatoms with the Richelia symbiont, and you have this Crocosphera single cell. And then you see them visualized here. Here we measure the 15N uptake, and you can see from the color coding, warmer colors means higher uptake, colder colors means little to no uptake, that there's difference between the different species. Trichodesmium, some filaments are active, not very active, but active, others are not active at all. The diatoms appear to be always active, very active actually, and the Crocosphera, you have a hard time often to detect any activity in the background. If you then get at iron, all hell breaks loose. So again, just for those of you interested, so the names of the symbioses are shown here and of the main players, and I've put some pictures here to the side to just so that you know how they look like. But it's not so relevant in the end. It's messy, it's going in all directions, and actually that's the message of the slide. The intriguing thing is that I said that at station 4 we didn't see any stimulation of N₂-fixation, significant stimulation of N₂-fixation when we added iron. However, when we look at station 4 here, at the diatom that has Richelia in there, there's a significant increase in N₂-fixation rates when we add iron. It's a really big one, actually one of the bigger ones we've seen so far. How is that possible? We don't see that in the book. Well, that's possible because it's being compensated by the fact that Trichodesmium, when you give them iron, stop fixing nitrogen. So what we're actually seeing, and this you can follow around for the other groups as well, look at the Crocosphera, they also start doing less nitrogen fixation. So in fact, when people say iron additions do not lead to anything, when you do these experiments with a bulk sample, I add iron and then I don't see any significant change and say, so the community is not responding to iron because they have more than enough iron, that's not true. It's an outweighing of some go more active, others go less active, and the result of it is that we don't see anything happening. And I think that's very relevant, and that might be happening at many other levels as well. So all the bulk data that we also generate and use is hiding a lot of stuff that is happening there. The reasons for that, one can speculate on it. I think in this case, why iron is reducing productivity is not toxicity, because we only added 2 nanomolars. It is also immediately oxidized, but probably that's the problem. It immediately gets oxidized and it coats whatever particles are around there, and it acts actually as an active chemical trap for phosphate. And probably what happens is that these organisms start running out of phosphate. Some of them probably are better at using organic phosphorus or have phosphorus reserves, and they can keep on going. So why do we care? Why should we care about that? It is because if we want to make predictions, we need to know who is responding and how they're responding. More uncertainty is actually added to this whole picture because I'm only talking about the cyanobacterial N₂-fixes, but the N₂-fixes out there in the ocean, they're much more diverse than that. So this is a tree showing the three main clusters of NIF-H, and the NIF-H gene containing organisms. NIF-H is encoding for the electron transfer protein that is involved in the nitrogenase reduction. It's characteristic. You can use it as a biomarker for nitrogen fixes, and in particularly this cluster 1, 2, and 3 have been shown to contain the nitrogen fixing organisms. There's some in other clusters as well. There's a huge diversity of clusters being elucidated recently. But if we look at these three clusters where we know that the main nitrogen fixes can be found, we see the cyanobacteria over here, the organisms that I already talked about before. But besides that, there is a huge diversity of other organisms of which we don't know what they are. And I particularly would like to draw your attention to this. This is a work done by Miriam Filippi, who was a PhD student in our lab, and she found this, what she called marine environmental cluster 1 and 2 nitrogenase, NIF-H gene sequences that belong to an organism that we don't know what it is. We thought it was a gamma proteobacterium because it was always described as gamma A. It's one of the most abundant NIF-H that you can find out in the ocean. The only thing I can say from the data we have, it's not a gamma proteobacterium. It's probably not even a coherent group of organisms. So we want to see how they actually, what role they play in the nitrogen cycling and maintaining productivity in the ocean. And for that, we cannot work with cultures. We have tried it. We cannot culture this, and many others have tried it. So we go for environmental procedures again, where we try to do that, what I showed you before with the NanoSIMS, and try to combine it with deep sequencing. And we did some of this work already in Lake Cadagno. It's in a meromictic lake, not so important, where we looked also N₂-fixes. In this case, we have a community of anoxygenic organisms that are sulfide oxidizers, that are phototrophic sulfide oxidizers that are growing in the sulfidic waters and that can fix nitrogen. And we wanted to know who is responsible for that. Actually, at first, we didn't find any of the logical ones. There were no cyanobacteria, so we did sequencing. Then we found that quite a few of the purple sulfur bacteria contained NIF genes. And we got nice mags. So what we then subsequently did is we used the 16S information to design fish probes, and then we could visualize them. Then we measured them on the NanoSIMS, and you can see all these purple sulfur bacteria are fixing nitrogen. But the wonderful thing about NanoSIMS and using this tracer technique is it's quantitative, because now you can start answering the question, who is actually responsible for the high rates, and there were really high rates of N₂-fixation that are happening in this particular lake. And in this case, it is this organism, Chromacium okeni, of which we didn't even know that was capable of fixing nitrogen, that is responsible for 80% or so of the nitrogen fixation that happens there. And this approach we would like to move to the open ocean also to target the MEC. So some of you might say, what an enormous amount of work for elucidating something like that, but it's highly relevant. Because N₂-fixation is actually what fuels CO2 drawdown. Whenever you see the chlorophyll pictures that I showed you before, I said the highest productivity is happening there where you find nitrate. That's where you have the highest growth rates as well, the highest productivity. But that's not where CO2 is being drawn down, because this growth is actually stimulated by upwelling of nutrients. Nitrate is being brought up, but with nitrate comes CO2. It's a absolutely not effective system to bring CO2 down. For that, you need actually a system where the physics are in favor of you and where water masses move down. And that's what you actually find is in these oligotrophic gyres. Water masses are being moved down with the organics in it. They're transported below the thermocline, and there the organic matter is being decayed and releasing CO2. For those of you who are interested in the biological pump, that's the biological pump. And then you can bury it for roughly 2,000 years and the CO2 will be coming up again. But if you then want to know who is responsible for this drawdown of CO2, actually there's a nice model paper by another group, Wang et al., that showed that up to 50–60% of the carbon drawdown that is happening in this environment is fueled by the nitrogen that is coming from nitrogen fixes. So it's a wonderful paper, it's a wonderful system, but it has one problem, because if you want to use this to predict what is going to happen with global change scenarios, the problem is that the iron requirements that are in this model are coming from one model organism that was in culture at that time, and that's Trichodesmium. And I showed you what happens when you give iron to Trichodesmium. In culture, it might maybe behave nicely and go and produce more N2 or fixed nitrogen, but in the environment, what we see is that they actually stop fixing nitrogen. So, and not only that, complexity is much bigger. The complexity of organisms that are out there is very high. If you really want to see what iron is going to do on nitrogen fixation in the future, you will need to know who is there, in which physiological state they are, and what actually is happening to the water column itself. And why iron? Has that anything to do with global change? Yes, it does. So, because dust imports are predicted to go up as well, for a simple reason that warmer temperatures lead to desertification. More deserts means more dust import. And so a simple prediction that was made is that means also more N₂-fixation. We're not so sure about that. Actually, we could have known that because we made a compilation of all the dust data that was there before and put all the measured nitrogen fixation rates on top of that. And then we always focus on this and see very high N₂-fixation rates there where we have high dust input. We should have maybe looked over here because you have similarly high rates over there where there's no dust input. So I don't think it's that simple. Not only will there be more dust input, actually, due to physics, and this is the last slide, due to physics, actually warming up the water column leads to more sluggish circulation, and more sluggish circulation will lead to an expansion of these Gyres systems, of these systems where CO2 is being drawn down, actually of these environments where N₂-fixers can grow. And this is not a model. That's the worrying thing about it. This is actually observational data from NASA. And it shows you the change in the size of these Gyres systems from 1998 to 2006. And you can see that these Gyres systems have been extending, I think they're estimated by 20% since in the last decade, and probably more. So what does this mean? More space for N₂-fixers and therefore N₂-fixation. That would be good news. More nitrogen is there to take down more CO2. Maybe supported by more iron input. That would be great. I mean, we have more dust input. So maybe more CO2 drawdown. Honest to God, I don't think so, because there's more complex things in there, and I'm not going to talk about it, but we have data, for instance, showing that the utilization of organic phosphorus, so the recycling of nutrients, will grow up if you have a system like that. You will be more dependent on organic nutrients for growth. And if that happens, one of the nutrients that's being used is metal phosphonate, and metal phosphonate, demethylation of these metal phosphonates will actually release methane. It might actually be that you can see a detrimental effect of the expanding Gyres systems. And what I wanted to have actually as a drive home message is that what we have a problem with currently is that these models, these global models that I'm talking about, are good at chemistry, they're good at physics. There's very little biology in it. Mostly there's only chemistry in it. There's some of them that have a bit of biology in there, but then they have one heterotroph, they have one phototroph, and maybe an N₂-fixer. But all of them do their discrete reaction, and there's no interaction with them. And I think these models are off. These are the models that we ever look at, and all of us show, also when you talk about CO2 emissions, I think they're wrong. For the simple reason that you cannot do a model like that without actually taking into account that you have interactions by microorganisms. And I think in that respect, this workshop is very good. And I think these models will have to include that to really be able to predict what is going to happen. I think on the short term, they will work, but on the long term, we will have massive changes that cannot be predicted without putting more complexity in these models. So I would like to thank you for your attention and apologize for talking too long. And I would like to thank this funding agency and also supporting institute and collaborators. And thank you once again. Thanks.
Thanks for that beautiful talk. Any questions?
Thanks a lot. I have a question on the first part. You're letting Microbial, they have sophisticated mechanisms. How is that sophisticated mechanism so that cheaper bacteria that do not fit into will not also have access to the Seagrass or to the product? and the bacteria will secrete granulation factors and signals to punish in this process.
Yes.
Do you think there is an equivalent in this seagrass come up with the bacteria in that?
So we're pretty sure. I mean, they definitely have quorum sensing. So there's a lot of. I mean, there's an overview paper about the rhizobia and pathogens that live in roots, and they try to genomically distinguish them. It's impossible. So the problem is, I think, the organism that we have. At first, we thought it was a pathogen because it looks like genomically like a pathogen. It has many of the things that you can find in pathogens, but it also has very many similarities to the rhizobia. What we do see, however, is that when they're around, there's not many other organisms in the roots. So actually, in general, there's not so many organisms in the root. They dominate the system, but not always. And that's a case that we're working on now. We see in one case a Vibrio taking over. And that's a thing that we, like doing one summer campaign, we see that Vibrio is becoming it. But they're also fixing nitrogen. So we don't know if that's purely pathogenic or if this is something that is going in there. But the complexity of the system, we don't know if it's similar. We're looking into that now. We have actually isolated the organism, and we're working on that as well now. So,
N₂-fixation is of course very important, but also terrestrial plant.
yeah.
I was wondering if you could speculate, know about why plants have never evolved nitrogen fixation by themselves. Why do they rely on extracellular symbionts? Why did they not come up with that?
That's a very good question. Actually, I do not have the answer. I mean, I guess the resources, it doesn't pay off. I guess it doesn't pay off to invest in these resources because it's not always needed. I mean, and in many environments you have changing, even in the Mediterranean where I showed you before, you have the system in spring, there's enough nutrients around, and you can then, if you have then a symbiont, instead of having the nitrogenase yourself, you can just eat them probably. That's, I guess, what they're doing. Get rid of the bacteria, and then when you need them, you let them grow up. You can. I think it's maybe easier to control and cheaper. That's my guess, but I do not know. That's something that we have not been looking into. Yeah. Yeah. I think it will be very difficult to predict because we don't. The problem is with all these models, of course, we extrapolate from that what we know. And we're going to a state which we do not know. I mean, we have everything happening at the same time also. I mean, we have a warming of the ocean. I mean, I didn't say that, but 90% of all the heat that is being produced due to the additional CO2 that is in the atmosphere due to oil combustion, basically, is going into the sea. The sea is warming up. The pH is going down. You have, at the same time, massive, I mean, one of the biggest effects is this Haber-Bosch nitrogen that's being put in. And we put everything at the same time. If you wanted to do an experiment, you would never do that this way. And we're doing all these experiments at the same time. And I think we really have a hard time to predict what is going to help. I don't think that you will ever to get the complexity in, or never, I'm not going to say. In the nearer future, will not be able to get all this complexity in, but I think we would need to get this complexity at least to a level to say, what can be the upper limit? Being Dutch originally, I care about whether or not we're off by 5 to 10 degrees or so, because that would mean that the Arctic ice shield starts melting and the dikes have to be built maybe 100 meters high instead of 20 meters high. And that's the kind of information I think you could get out of there. I'm not sure that they're going to be better to predict. If you put the complexity in there, better to go and predict what is actually going to happen, because I think the models will not be able to do that. But it can give you an upper rate, and isn't that important actually to know what is the worst case scenario, because you will have to prepare for that. And I think, and then you can say, okay, it's not worthwhile to prepare because it's too expensive, but at least you would need to have some worst case that is maybe more realistic than the worst case that they have now. Because if you look at these models, the variation that they're talking about is very small, and that is because all the effects that they have are basically linear. You have like more nutrients, more productivity, more CO2 drawdown. There is no possibility that you give more nutrients and first go up the productivity and then it crashes. That doesn't exist in these models. But in the models that I've been seeing around here, this is actually what happens in reality. And I think that's something that is missing in these big models. The modeling guys that do these big models hate that, of course, because every line of script that they have to put in makes these are already crazy to run as they are just for the physics. But I think at the moment it works because we're still close to the situation as we know it. But the further we go away from that situation, the more CO2, whatever, that the worse it will get. Sorry for this long answer, but I get emotional when it comes to that. Yeah.
We'll take one last question.
Very technical one. So how long did you incubate the organisms? So you showed that they responded negatively to iron addition.
Yeah.
What was the incubation?
24 hours. We try to always keep the incubation short. So we do 20 hours to 48 hours because we don't want to have the effect of that we start growing up something new. And that also, that I should maybe say, because when I said like the nitrogen limitation that I said before, I think that's also not that straightforward because we did experiments in the South Pacific Gyre where we added nitrate, ammonium, and nitrate and ammonium and looked at what happens. Nothing happens. Because the community there is not limited to nitrate and ammonium because they never see it. So, yeah.
Includes the day-night cycle?
Sorry? Yeah, that's for sure. Yeah, we do it, we incubate on the deck, we make sure that it always includes at least 1 day-night cycle. Mostly 1.5 to 2 cycles are involved, yeah. But then we want to stop because then you get all kinds of bottle effects that we don't want to get.
All right, that's great. So let's leave it there and let's thank all.